Dr. Orchid Garcia, who's currently a research fellow and lead for bioprinting and tissue regenerative technologies at Johnson & Johnson. In this role, she is responsible for evaluation and execution of technical strategies and new technologies integration to develop a new class of next generation healthcare solutions. And today, Orchid is going to talk to us about how bioprinting is evolving how we create and deliver personalized products and solutions to treat patients. With that, Orchid.
Thanks, Rachel. Let me get my slides. There we go. Hopefully everybody can see okay.
It looks great, thank you.
Perfect. So thanks for the kind introduction and the invitation to address everyone today. I'm very excited to speak to this team in particular because I definitely see a lot of potential here for collaboration, not just on product-focused technologies, but on technologies that will accelerate product pipelines and provide some strategic and deep insights into how we innovate at Johnson & Johnson. So as Rachel mentioned, I'm a research fellow at Johnson & Johnson. I lead the technology vertical lead for bioprinting and tissue regen technologies. What folks may or may not know is that a few years ago, Johnson & Johnson made a strategic investment in 3D printing. They made the investment in various different channels. So we have a metals channel, a polymer channel. We have a scan to health and point of care, which is focused more on bringing this technology out of a manufacturing environment and into a healthcare facility. We have sensors and obviously bioprinting. The reason that this investment was made was the foresight on the part of folks at Johnson & Johnson to realize that technologies like 3D printing and bioprinting are going to become the norm in healthcare moving forward. So this lives into our model of behaving like a 133-year-old startup. And so I'm excited today to share a little bit of why we made this investment in bioprinting, specifically what we're looking forward to, where we're focusing our efforts, and where we potentially see opportunities in the future. So one of the reasons that J&J made this investment in bioprinting in particular is there's various challenges that we're seeing in the current healthcare environment, especially now post-COVID. You know, prior to COVID, clearly there was a focus on outcomes, scrutiny on cost of care. We're moving towards more patient-specific care rather than the generalized medicine paradigm that we're used to. We have aging, unique, and vulnerable populations. But on top of that, challenges like COVID, which are most top of mind at the moment, present new and interesting challenges that we feel that some of the technologies that we're investing in definitely have a role to play in helping live into this new healthcare paradigm, which may become a distanced healthcare paradigm. So some of the strategic investments, as you may be aware, that J&J is pursuing are in the areas of robotics and digital surgery, ultra personalization, improved diagnostic tools, and predictive modeling. Now, I may be biased, but I feel that bioprinting is the nexus of where all of those technologies meet, and there's definitely a lot of opportunity to leverage bioprinting in all of these different channels, and I'll kind of walk you through what that looks like. So in terms of how we view bioprinting internally and how I describe it to my colleagues, because it is a bit of a mystery to folks. It is, you know, a bit of a nascent technology from the industrial perspective as well, when you're looking at scale and supply at the levels that J&J operates at. And so the best way that I like to describe it is it's the next step in evolution of ultra-personalized medicine. So, you know, clearly we've started out with intuition-based medicine. Patients come in with signs and symptoms. A physician will make an educated guess based on their experience, based on their education, as to how best to treat this patient. And there may be some trial and error, but that's typically the paradigm that we see in healthcare. Now, clearly, we've moved into evidence-based medicine, where treatment is based on large clinical data sets, which are generalized, and so the ability to generalize those to a specific population, a specific age or disease state, has kind of moved us into the precision medicine space, where we're making decisions based on big data and algorithms. Moving one step further, we've got the personalized medicine space, which is what we see a lot of now, the genetic tests. You can see what your nutrition profile looks like, or what genes you may be carrying, etc. Those predictions are based on static patient, genetic, environmental, lifestyle factors that are given as an input, a snapshot in time into where a patient is at in their health journey. Now, with the evolution of ultra-personalized medicine, bioprinting kind of fits really nicely in there, because we can base treatments on dynamic individual patient needs and biological status at the moment of treatment, but also based on a lifetime of genetic and epigenetic changes and how those genes are expressed, et cetera. And so we can do things like take patient-specific cells, create an in vitro organ-on-chip construct, screen against various drug targets or potential therapies, and look at which makes the most sense for a patient. So what are the applications? I often get the question about, 3D bioprinting is great. However, how are you going to ever get a product to market? And in the interim, while a lot of regulatory agencies are refining guidance documents and putting that path to market in place, where can we generate value from this type of technology? And there's clearly two different buckets in my mind. One is I like to refer to as the product supportive technologies. So I lovingly refer to those as my innovation accelerators, and those are bioprinted constructs that will actually accelerate the pace of innovation or a pipeline of innovation for other products. So we can reduce reliance on animal testing, which can, clearly be less than ideal models for what happens clinically in patients, and can lead to a lot of delay in the availability of various treatments, because you have this iterative cycle where you take a drug or a device into clinical trials and it doesn't necessarily perform as it had in an animal. We can improve in vitro test methodologies to make them disease-specific, patient-specific. Very often when some of our teams are looking into R&D, into new treatment modalities, it's difficult for them to order tissue or cadaveric specimens with a specific pathology. You get what you get. So, sometimes they're flying blind in trying to telegraph how a technology will actually link up to the disease state in the end. Clearly improved in vivo modeling, so the ability to plan surgery, the ability to print constructs that more accurately replicate what happened in vivo, and be able to more efficiently hone in on what some of those targets, some of those therapeutic targets might be. On the other side, we have the transformational product technologies, very much similar to what we've heard today in regard to some of the corneal constructs. And these are those product technologies that are going to transform medicine and create new treatment paradigms. So technologies that regenerate and repair rather than replace. And very often in medical device design and in treatment for various diseases, we've been agnostic to regenerative medicine, and really bioprinting has the opportunity to take that therapeutic challenge to the next level and create a space where we can not just treat a disease, but potentially regenerate some of that tissue, restore function, etc. So this is just a case study, and I won't walk everybody through everything in painful detail. I know it's a bit of an eye chart, but when trying to communicate the potential immediate impact of bioprinting to therapeutic discovery, I like to use the in vitro tissue screening model. We've seen a lot of these. recently. I think there was an article in the New York Times most recently about the team at Wake Forest looking at bioprinted tissue constructs for various COVID testing applications. Clearly the traditional paradigm is what we see on the top. Pre-discovery, when we're screening large drug libraries, we're using static 2D culture models. Clearly there's a wealth of data that demonstrates that these types of models fail to replicate the etiology, pathology, disease progression, etc. There are various architectural cues in a lot of the diseases that we look at, and those are clearly missing from a static two-dimensional culture where you don't have the appropriate polarization, architecture, etc. Bioprinting allows you to create that three-dimensional structure that will allow you to not only look at a cellular molecular level, but also the system as a whole and how that influences the initiation and progression of various diseases. So as we move into kind of, pre-discovery to drug discovery, and we're honing in on targets, or we're honing in on devices that we're looking to move to, preclinical and hopefully clinical trials, some of the monolayer and multi-layer systems also, again, fail to recapitulate what those cues are. It's also difficult to incorporate things like microfluidics, which may be helpful for various transient biological cues that we may want to replicate. So, very often in traditional drug discovery, you move through this cycle and you get into a preclinical animal model, which is great for recapitulating various mechanisms, but not the entirety of what's happening in the human system. And so, animals are unique. Mice have things like very long telomeres that allow them to regenerate in certain situations. And so the one-to-one correlation isn't always there. There's a lot of guesswork involved, and the data set that you are looking to take into a clinical trial isn't always complete. And so many times what you've looked at in a petri dish or in a tissue culture plate may not play out in a preclinical animal model, and you may have to make multiple iterations to see, to find a target, to find a device that actually addresses what you're trying to address. And even then, when you move into a clinical trial, you may be faced with that iterative cycle again. So, age, disease progression, various genetic factors, demographic factors, etc., may affect how something that looked really good in the preclinical sense doesn't necessarily make it and meet that mark in the clinical trial phase. So all of this kind of creates a storm of an imperfect data package sometimes that we then, go to regulators with. And it may be difficult for them to telegraph how this may perform in a larger real world setting. So when you're not dealing with, the physicians who are trained to the levels that they are in various clinical trials, et cetera. And so that's why you kind of see this iterative cycle and you see this drawn out, and folks get very frustrated because the availability of certain treatments isn't quite there, isn't happening fast enough. But it's because this process is an imperfect process, and trying to capture and fill in all these gaps requires that iterative process. Now, I firmly believe that bioprinting has the opportunity to accelerate a lot of these processes, and not completely replace, but act as an adjunct, and even in some cases potentially replace. So, high throughput, three-dimensional, architecturally relevant models can be used for screening drug targets initially, that will allow us to focus in on the most efficient, potentially, best, suited drug candidates to move through some of the later iterations of the discovery cycle. When we're looking at these models, we can then in vitro, without using animal models, manipulate some of the parameters that we think may be relevant to the disease. Various microfluidics, the addition of various cytokines or molecular moieties that we feel may be dysregulated in various disease states. Not only does this provide us a better model, It allows us to hone in on that very specific mechanism that we feel may be driving a certain disease state. We may not be able to get that from cadaveric tissue. Additionally, for ethical concerns and other issues, animal models are not the ideal, and so potentially we could replace those models with better models that are architecturally accurate, et cetera, that give us better insight into how these drugs and devices might perform clinically. And then again, in clinical trials, if we have a better vantage point of how these devices and drugs may perform, we may better design our trials. So, look at the appropriate populations, look at the appropriate disease progression, et cetera, and provide, at the end of the day, the goal is to provide regulators better data sets, better foundational models, et cetera, for their approval, so that they can feel confident in the drugs and devices that they're approving, and we can bring those drugs and devices to folks a lot faster. So how are we at J&J looking to incorporate bioprinting now and in the future to kind of change the trajectory of health for humanity, which has become our mantra? Right now we're really focused on the disruptive innovation piece. So looking at pioneering these game-changing solutions that have the potential to accelerate various treatment paradigms through the R&D cycle, which is, I just walked you through that one case study. So in particular, things like new materials, new processes, potential products, supportive technologies, or in vitro tissue models. We're really not focused on the hardware piece of things. I don't see us getting into the capital equipment game. But it's those patient-focused solutions. How can we better treat patients? How can we bring new healthcare solutions to them? How can we deliver our healthcare solutions to them better? The next step in my mind, obviously, is the personalization piece, which I talked about earlier. So translating something that could be very generalized into something that's very specific to improve outcomes for specific patients, not even specific patient groups, but specific patients. So implants that are matched to the patient rather than an implant that's a general size and put in a patient. Specific, in vitro screening and planning, either surgical planning for difficult cases or, in the medical treatment side of the equation. And then finally, the access piece is something that we're looking forward to on the horizon. So as we're moving into a socially distanced society, we are going to have to anticipate, distance medicine as well. Initially, the challenge was to bring some of these healthcare solutions to folks who don't necessarily have access to world-class facilities like many of us are lucky to have access to. But now we're looking at how can this play into the new distance paradigm where patients and physicians may not want to have that face-to-face contact. You may need to deliver healthcare solutions in home. So how do we deliver some of these healthcare solutions in a distance manner? Is it point of care delivery? Clearly there are going to be, there's going to be the need to set up infrastructures for on-demand solutions, et cetera. So how are we doing this at Johnson & Johnson? So clearly we've set up an internal team of experts that have these competencies, have these skills, but we realize that, if we do all of this internally, we're essentially innovating in a vacuum. There are some freedoms that academia has versus industry when, chasing some of these new mechanistic paradigms and some of these new product solutions. And so we've strategically, not just in my group, but also across the organization, set up various partnerships and are always looking for tremendous partners to be able to work with, to understand the technology, develop it, steward it, et cetera. And so, we have strategic partners. We've got various partners in industry and academia that we work with. We've got technology partners, various startups, et cetera, that we're either working on targeted application or platform technologies with, and then clearly the hospitals and medical centers. So the folks at, who are on the front lines, who will be using these types of technologies, and, who, if these aren't designed properly, will not have a use for them. So we've partnered across all of those channels. My team, and specifically my front-end R&D lab, is located in Dublin because we have a wonderful partnership with the Amber Centre in Ireland, which is a consortium of colleges and universities focused on materials development. So Trinity College is where we're based, but we have a collaboration with the Royal College of Surgeons, et cetera. We also have a partnership, which was announced earlier this year, with TNR Biofab in Korea to create various platform technologies based on some of their previous work and some of the internal work that we had previously done. So, the partnership model that we've embodied, to me, is refreshing, because this is where, the nexus of all of these great minds come together, and we can move some of these advancements forward faster. Now, the piece that we're missing is the research and space piece, which I think will probably be the lowest hanging fruit from my perspective, but being able to gain some of those strategic insights, or some of those insights, and leverage this technology to get there, I think will help us build a foundational data set for some of these later stage product technologies that we're looking forward to bringing to medicine. So, what does the future hold for 3D bioprinting? Clearly I'm biased, but I feel it's a key enabling technologies that can address a lot of these challenges that we talked about earlier, and some of these new challenges that are coming up. Clearly there's a scrutiny on cost of care. When you're talking to hospital systems and hospitals, there's, there are committees that will make, treatment decisions based on cost. And so, being able to source biomaterials from consistent, reliable, scalable supply chains will eliminate the need for human sourcing and processing, which will bring down cost, also bring down a lot of regulatory implications. Improved outcomes clearly is one that we discussed. So, to be able to regenerate rather than repair, to restore function, not just mechanical, but biological function. And being able to consistently and reliably provide these predictable outcomes across patients is an important one for us as well. Finally, a lot of these treatments that are regenerative, that are sourced based on various allograft and xenograft technology, are limited based on donor supply and donor quality. And so we never want a patient to not have the option for the best available treatment because there's not a donor available. Agility and responsiveness clearly is a big one for us. So being able to use this technology to provide on-demand flexibility, tailoring all of these treatments specifically for patient needs. Multimodal delivery is one that I'm probably most excited about. I think bioprinting lends itself quite nicely to different delivery paradigms. So for example, you can have bioprinting in a traditional manufacturing environment. However, point of care, so manufacturing within a healthcare facility, or even in situ, so in the operating theater. I think this is where bioprinting will probably make the biggest strides in the near future, is some of those technologies that bring the larger technology of bioprinting into a patient care setting, I think will be most impactful. Targeted solutions, again, minimizing risk by tailoring treatments specific to various patients. Expanded access, again, minimizing regulatory hurdles that may be encountered with other types of treatments. And then ultra personalization, which we discussed earlier on. So one of the things I was asked to kind of reflect on are what are some of the gaps and challenges that we're facing and that I'm seeing on the industry side, and these may be very different from the academic side or from the research side, but clearly there are gaps and challenges in standards for medical and biological additive manufacturing and the maturation piece. So, bioprinted constructs are very different from a 3D printed metal or polymer construct in that you print a metal construct and you ship it to a location and you can trust that it will essentially be, unless something, terrible has happened, it will essentially be the same construct that you printed. Now for bioprinting, that's not always the case. Some of these technologies don't lend themselves very well to transport. So how do we guarantee the quality and integrity of not only what we printed in the lab, but what was matured over time and what was transported and delivered to a patient? Specific guidance document for 3D printed devices containing biological components. There are some regulatory agencies who are being very forward thinking and reaching out to us and having these discussions, along with having discussions with academics about how best to develop these technologies and what we see on the event horizon and how they can adjust to suit those needs. And so, those guidance documents are forthcoming, but for scientists like ourselves, it's not fast enough. Fit for purpose regulatory framework for personalized devices and products. So for those one-off, printed on site, printed specific to a patient, printed specific to match an anatomy, those regulatory frameworks are still lacking. I think that we've done a great job in being able to leverage what's currently available right now to kind of get closer to the fit for purpose piece, but I think there's definitely an opportunity to, engage there and create a more robust mechanism. Again, a manufacturing framework, manufacturing and delivery framework, scale and supply capabilities for biologic and cellular components. So clearly, cells are not able to be endlessly expanded. The biologic sourcing of materials to ensure quality, integrity, reproducibility, all of that kind of stuff. There are some gaps. Data and intellectual property protection, if we are making patient-specific constructs, or if we do have patient-specific genetic and epigenetic data, how do we manage that? And again, on the opportunity side, clearly what I mentioned, there's plenty of opportunities for regulators, academic, government agencies, industry to engage and work simultaneously to develop some of these frameworks. I definitely think there's a huge opportunity to integrate, the automation of AI, robotics, digital surgery, and not just for the creation, but scale, supply, delivery, et cetera. So again, bioprinting is rapidly evolving. J&J has placed a strategic bet because they realize we're headed in this direction, and we think it's important to understand this technology, develop it, because clearly this is going to be what our patients are going to be demanding in the future and what healthcare needs in the future. So we're definitely looking forward to seeing the field progress, engaging with individuals like yourselves to see how we can make strides forward. And in closing, again, our mantra is always to have the patients in our forefront and look to change the trajectory of health for humanity. And clearly, in my mind, bioprinting really suits that entire mantra. So I will stop there.